Well, Nick, a member of our carnivore certification program. Not quite certified yet, but working on it. How have you been?
Yeah, I've been great. And how are you
Yeah,
going,
yeah.
Andy?
Yeah, pretty good. Busy as usual, but yeah, I'm making sure that I'm looking forward to this conversation because you have a history of engineering. And there's a lot of parallels when it comes to these systems of the body and then systems that we build. In fact, most of the systems that we build actually directly mimic what is occurring in our body as observed. So I'm looking forward to this because I think that you and I have very similar mindsets in that regard. So yeah, but before we get into that, give a little bit of your background. I know that I just said that you have a little bit of background in engineering, but just for people to understand, more about who you are and the way that you think and what got you into, wanting to learn more about carnivore and systems of the body and all that.
Okay, yeah. So, I mean, you said engineering. So I did study engineering at university, back when I was 18, 19 but by the time I finished the course, I wasn't interested in becoming an engineer.
Right.
And
Yeah.
I was sort of heavy. I think I was spending more time in the gym than in classes at university the last couple of years. And from that, I got into the fitness industry and became a fitness instructor. This was the early 90s, so it's a while ago now. And for the next 10 years or so, that's what I was pretty much doing. Um, uh, Towards the end of that, I sort of wanted to sort of make more money. There wasn't a lot of money in fitness back then, and I got offered a job in IT and fell into that. So for the next, well, 20 years after that, I've been working in IT. So I went from jumping around all day to sitting around all day, and my health and fitness sort of slowly declined over the years. I ended up getting married late and having children later in life compared to most people, and I was sort of turning 50, and I got two young kids with maybe another one on the way, and I thought I want to be able to sort of play and jump around and keep up with my kids, and um, started saying, okay, I had a background in this, I can get back into it. And I started looking around online, like what's the best thing to do, especially from a diet perspective, because I was following, I suppose, the food guidelines, if you like, and really interesting. Like I never went down the path of becoming vegan, but I sort of build this mindset that, oh, we eat too much meat. I've got to cut back on the amount of meat that we eat, and eat more vegetables. So I went to eating meat maybe two or three times a week, and
Mm.
it didn't work. Obviously, it's like I was getting lazier and bigger and less healthy and all sorts of stuff. So when I started, okay, I'm going to look into this properly, I looked into vegan, I looked into all sorts of things, and the thing that sort of got me originally back then was becoming keto. So I started
Mm.
doing keto. I did that for probably a year and a half, and through that, and again, reading more, and trying to refrain from using the word research because I'm not a scientist, but just from reading and podcasts and everything I came across, the usual suspects, mainly Bart Kay and Sean Baker and who was it? And Anthony Chafee, of course.
Yep.
And
Yep.
I think, okay, that sounds interesting. That sounds interesting. I should try full carnivore. I should try. And I sort of, as I was doing that, I was becoming more and more, I suppose, leaning towards ketovore than keto, because I started off dirty keto, making all the almond bread and all
Mm.
that sort of stuff. And then towards the end of that, I was more clean keto. So it was basically, meats and a few vegetables. Um, and then I thought, okay, it probably don't need to do carnivore, but let's try it anyway. And it's a bit over 2 years ago now. I tried it, and for the first, I said I gave myself a month. I'll do it for a month. If there's no difference, then I can go back to eating a few more vegetables. And I think it was about week three. Um, I woke up one day, and it was almost like an overnight thing, like 21 days, so 3 weeks after dropping all vegetables. I felt 10, 15 years younger, or I felt like I did in my mid 20s. And it was just, wow. Like, okay,
Yeah.
So I did the month and I thought, okay, well, let's do it for another month and let's do it for another month. And, here we are 2.5 years later and,
Wow.
95, 97%. I still have some coffee and occasionally, sneak something else in. But for the most part, I've been carnivore for 2.5 years and much to everyone's like shock and horror and
Yeah.
everything around me. Um, my wife and kids, they're not, I suppose, full carnivore, but I tend to cook breakfast and dinners. So their breakfast and dinners are steak, bacon, and eggs, or some
Good.
variation thereof. And lunch, the kids usually have something a little bit off carnivore for lunch. So, but yeah, I feel, you know, better, stronger, faster than I have since I was in my early to mid 20s. And yeah,
Yeah.
I've learning more. I mean, I religiously watch your channel, Bart K. You and Bart K are my two favorites. Like every time there's a video, I almost like I actually get excited when there's a new video on because it gives me something to sort of delve into. I try and watch a few of the others as well, but there's limited time. I still work. Got
Yeah.
three young
Yeah.
kids and all that sort of stuff. So yeah, it's
Yeah.
I did yeah. So last year, actually, based on your recommendation, I got in touch with Coach Peter, and earlier this year, I joined the Carnivore Coach course. And I've gone through everything. I just haven't sort of said, okay, let's sit down, review everything, and then do the test. So I haven't done that yet. And then I also joined the fitness certification one as well to sort of renew my certification in fitness.
Right. Yeah, well, this sounds like your redemption arc here in your life. Getting back into the activity and making sure that you can reach your most optimal health and get back to your health like you felt in your 20s. So that's good. You've been carnivore longer than I have. I think I've been. Oh, what? I mean, this is maybe we've been carnivore for around the same time. I started in August of 2022, actually. So
Yeah,
yeah, I mean,
there we
no,
go.
no,
Me,
it's
me
yeah,
too. August.
yeah.
It was my birthday.
It was around my birthday.
My,
Yeah,
yeah, I
yeah,
started on the day after my birthday in August 22.
yeah. So I mean, okay, so around the exact same time. So all right, cool. Look, we have more in common. Yeah. But So I actually I want to apologize. I said that I think I may have said at the very beginning you were an engineer, but no, you're not. You didn't go into engineering. So you went through the rope, so to speak. But then you
Yeah, I
went
did
down a
all
different path.
the theory stuff at uni, but I never
Yeah.
practiced in real life. So no, I don't consider myself an engineer from that perspective.
Right. But would it be reasonable to say that you sort of have that the mindset, not necessarily to want to go and be an engineer, but to think that way?
Yeah, I was always, the sort of kid that used to pull apart their toys to figure out how they worked, and
Right. Okay.
if I wasn't bored with them, I'd put them back together again. And whenever I got into anything, whatever it was, I wanted to learn more about it. How did it work? Like when I bought my first car. Okay, I needed to know about cars. So a friend and I, who similar mindset, we went and bought you know, from the wreckers, we got a wrecker that where they throw away old cars. We bought this old car for a few $100s and pulled it apart just to learn about it. So I suppose the same thing when I got into the gym and then health and fitness, I wanted to know more about how the body worked, and that's why initially I did a basic fitness course, but I jumped in further and I went and studied proper anatomy and physiology and a bit of biomechanics as well to learn more about it.
Mm-hmm.
Um,
Yeah.
and then I sort of moved on from that to IT and didn't forget about health and fitness, but sort of put it in the background.
Right. Yeah. Well, I mean, your questions that you said, the topics that your questions revolve around seem to fit that sort of way of thinking, which is why I asked that. You were wanting to ask some questions about, how muscle fibers actually like work, at least a deeper level of understanding of it, as well as some other aspects. Because I mean, at the end of the day, like I said, biochemistry basically emulates a lot of the systems of the body, emulate the systems, I should really say it's the opposite. The systems that we build, also, they directly emulate, certain elements of the body itself. Like, even just the way that our body makes ATP, the ATP synthase motor is literally a motor. It has a stator. It has everything there in a regular motor that we've built. So that's why I asked you that question. But yeah, well, I'm looking forward to your questions. I mean, you can ask them in any order. And fortunately, the topics that you've touched on are some of my favorites to talk about, and they were my favorite to learn like a year or two ago, or well, more than that actually, whenever I was looking into it. So ask away.
Okay, well, let's start with some of, I suppose, more simple stuff from the original carnivore course.
Okay.
Vitamin C. So from my understanding, vitamin C, we don't need, as when I say we, as carnivores, or I suppose everyone is a carnivore, but eating our proper diet, we don't need anywhere near as much vitamin C as people who eat a lot of carbohydrates do. And one of the reasons is that it's not fighting to get into the cells via the GLUT4 transport. But one of the other things that I've come across or heard is vitamin C is also used to convert non-heme iron to heme iron. Is that correct?
I believe so. It happens in the gastrointestinal tract before absorption, I believe.
And then I suppose
via a
from that
specific
is
enzyme. Yeah.
How much of a factor of not requiring as much vitamin C does that play into it? Like, what's a bigger factor? The fact that it's not vying for the GLUT4 transport, or we don't need it because, like.
Yeah. So I actually think there's an even bigger factor. I think they all have roles to play, obviously. So one of the things I want to say is that not only is glucose and not only are glucose and vitamin C a competition for GLUT4, but also through GLUT1 too. So there's actually, it's multiple GLUT transporters, and it's because vitamin C and glucose have very similar structures with respect to the aspects that the transporters actually identify, so to speak, whenever they encounter those molecules. And then we talked about, yes, the conversion of iron, elemental iron to heme iron in the gastrointestinal tract. But also there's one that I think, once again, is the more broad based level that I think is the most significant reason as to why carnivores don't actually need that much vitamin C, or human beings at the very least, which is vitamin C's role in collagen synthesis, and also its specific roles in amino acid metabolism. On the top of my head, I cannot remember the exact specific step, because whenever you look at like all of these biochemical pathways and stuff, it's hard to remember all of the intermediates and all the enzymes. It's like borderline impossible. You can remember you can remember like the glycolysis, like the prototypical ones and beta oxidation and stuff. But like when you talk about certain levels of amino acid metabolism, and when you talk about hydroxyproline metabolism and vitamin C being involved in that pathway, it's like, okay, yeah, sometimes they're going to slip your mind. So I don't remember at the very top of my head what it's involved in. However, we do know that vitamin C is tightly involved in collagen synthesis, but particularly whenever you're not consuming, or when you're not consuming very bioavailable levels of these amino acids and of collagen directly as well. For example, everyone thinks of vitamin C deficiency, they think of scurvy. Like that's really what it is. That's what scurvy is. And what people have a picture in their minds, like, I don't know if it's the same for everyone, but it is for me. I was always growing up like scurvy was the pirate's disease. It was the sailors, like, okay, they got scurvy because they didn't have fruits and vegetables, right? Well, not only did they not have fruits and vegetables, it was really the fact that they didn't have anything except like hardtack at one point, which is just flour and water. But one of the symptoms of scurvy that people think of is like bleeding gums. But what coincides with that is that their legs were all like black, like it was like their blood was pooling at the bottoms of their legs, and the reason for that was it was blood, but it was because their blood vessels, which are lined with collagen, there's different types of collagen. I believe it's type 3 collagen that lines your blood vessels, but I could be wrong on that one. Collagen lines all of your blood vessels, and whenever you don't have enough collagen that you're either directly consuming or that you're consuming bioavailable amino acids to synthesize, or really acquire through your food, You're going to need to depend on vitamin C to synthesize collagen endogenously. It's sort of like how we have to depend on our body to synthesize cholesterol endogenously when we're not consuming the thing.
Mm-hmm.
Well, yes, your body might be able to create all the cholesterol it needs. It doesn't mean that you're going to flourish that way. It means that you can survive. Well, in
Mm-hmm.
the case of vitamin C, you can't even say that you will survive without any of that. Like you need either one or the other. Like you either consume lots of, this is my opinion. You either consume lots of bioavailable protein, collagen, you consume all these animal products that allow you to not have these problems and allow you to not have to consume so much vitamin C to get the collagen synthesis that you require for base level health, or you depend on vitamin C. If you have none of it though, well, then you can expect things like bleeding gums, and you can expect your blood vessels to fall apart, which is the same thing. That's really what it all comes back to that. It's a lack of strength of blood vessels. They just start to disintegrate. And so I think that's actually the primary reason why humans don't require, whenever they're on a carnivore diet, they don't require not even close to how much vitamin C is recommended, which is like 70 to 80 milligrams per day, I believe. In the people that have been studied, the actual blood level that is required, the minimum level at the very least, to mitigate scurvy, I believe it's 10 micromoles, which I believe is like 1.3, maybe even 1.1 milligrams. But that also fails to even recognize how much you recycle, the genotype and the phenotype of the individual studied, because if they were eating a normal diet, again, like we were just saying, that could influence how much is required to mitigate scurvy. And so even according to those people studied, it seems that the dietary requirement, therefore, because of all this ability to recycle it, the different needs, the lack of utilization of vitamin C as a reducing agent, so an antioxidant, whenever you're not sustaining tons of ROS production or tons of inflammation. It seems that even according to those people, it's in the nanograms, billionths of grams. And then you
Mm-hmm.
also add on to that the ostensible lack of requirement, as much of a requirement whenever you're consuming lots of meat. I mean, I was just talking to Anthony Chafee relatively recently, a few weeks ago, and I said, I wouldn't say that I'm to the degree that I believe that vitamin C isn't a vitamin or it's a conditional vitamin. However, I wouldn't be shocked if we ended up starting to realize, like, oh, so you only need this much vitamin C as like a backup. Everyone wants to talk about gluconeogenesis and fat oxidation. Those are your backup sources of energy production. It's like, we already know that's nonsense. But in reality, you could make that argument for vitamin C more than you could with those pathways. Like you could actually, even if it's not strictly like 100% true, because again, I don't know if I'm on that level yet, if I ever will be. But, clearly it's not just a matter of carbohydrate competition, because you have people like the Maasai that drink plenty of carbohydrates from milk. And they don't come down with scurvy either still. And they're not eating loads of fruit.
They're also eating a lot of meat too, yeah?
Yeah, exactly. So, I believe that there's only like one source of, I think it's like spleen has vitamin C, right?
Oh,
I think
okay.
so
The liver has some?
potentially, yeah. I can't remember all I know that spleen, like if you're not, like you still, even with liver, you got only a little bit of vitamin C. I believe spleen is like...
All meat does have trace amounts, yeah.
Right, yes. All meat has trace amounts. I'm talking about in significant amounts. Like spleen has the most significant, I think, in like, at least in ruminant animals. I could be wrong. On the specific details like that, it's like, I don't know.
I don't think I've ever eaten spleen.
Me neither. I don't really plan on it, but...
It'd be curious. I do like liver occasionally.
Yeah, I've never had it either.
I never used to like it, and just on a couple of times that Anthony Chafee is talking about having it raw, I did try it raw, and it was actually quite nice and sweet. There's a sweetness to it, yeah.
The sweetness part actually doesn't surprise me, because sometimes like raw meat generally can have a sweet initial taste, I think.
Mm-hmm.
That's again, it depends on multiple factors, but I've heard that before. It's not a foreign claim. So
But the liver's where, stores a lot of glucose and
yeah,
glu
hmm
glycogen as well, and so
yep.
there might be it might just
Some
be all of that. So
Yeah, some. I mean, usually most of it's going to be in the fed state that it'll store onto lots of glycogen. So if you get like a fresh kill after an animal has been is well fed, then it'll have more glycogen in it. Um, the the more that the animal has time to be in a fasted state, your liver glycogen stores are what go first. So I mean, it makes sense that there could be there'll still be some traces, sure, yeah, but I mean, we're talking like trace.
okay,
Yeah. Yeah.
so yeah. Yeah, I mean, when you cook it, I don't really enjoy the taste when it's overly cooked, and it definitely doesn't taste sweet. But the raw
Mhm.
or very rare, yeah, there's definitely a sweet sort of
Yeah.
side to it. So going back to GLUT transporters,
Mhm.
how many? Is there 11?
I believe there's 14,
Oh, 14.
um,
Okay.
but the only ones that are actually involved in glucose metabolism are GLUTs 1 through 4. So, for example, GLUT 5, once you get to like GLUT 5, you're talking about like fructose metabolism. And for like, I also don't know if they even go in order, like GLUT 5 and then GLUT 6 and GLUT 7. Because from my understanding, it's like GLUT 5 and then GLUT 7 and then GLUT 9. I genuinely can't remember at the top of my head because they're very fringe in metabolism. Like,
Okay.
with the exception of GLUT 5, because GLUT 5, again, primary site of like glucose uptake, especially they're particularly on like liver cells for obvious reasons, because liver is the main site for fructose metabolism. But I believe you also have some GLUT 5 in your small intestinal cells as well. So there's some digestive cells that they exhibit, present with GLUT 5 on them. But yeah, I believe in total, just like GLUT transporters, all more broadly, you're talking like 14, I believe.
14.
Yeah.
And is GLUT4 the main one for getting glucose into cells?
Postprandially, typically, so after one eats, they will have, the typical person, they're eating a standard diet, so carbohydrates, fats, and proteins, and that's the most conducive way of getting your what we call I to G ratio really high. So your insulin to glucagon ratio. So just think of insulin and glucagon as yin and yang. That's really, just think of it, the
Hmm.
Taoist symbol. You get a very high I to G ratio. Insulin is actually what stimulates the what would you say, the translocation of GLUT4 from inside the cell to the exterior, to the membrane. So a lot of people immediately think, and the graphics online don't help, that GLUT4 is just embedded permanently into the cell membrane, like most other transporters are. That's not true. They actually have to be recruited to the membrane, and then they bud out. And there's things that stimulate them to move and open up. And insulin is the primary factor. It's the prototypical factor, the prototypical variable. It doesn't do it directly. Insulin binds onto the outsides of cells through insulin receptors, usually tyrosine receptors. And then it causes a chemical cascade of second messenger systems known as phosphoprotein phosphatases is typically what it's involved in. But what it does is it stimulates, ultimately, GLUT4 to the plasma membrane to take up glucose in sort of hyperglycemic situations or events. You've also got, though, the other primary factor, which is really independent of insulin, actually, which is AMP. So we always hear about ATP,
Hmm.
and you've also heard ADP, because whenever ATP is used, you've got PI, inorganic phosphate, and ADP. However, ADP can go through a second step, which is where it loses another inorganic phosphate, and then you've got AMP. So AMP is
Hmm.
always reflective of low energy supply, and usually like a dip even below the normal homeostatic set point for like maximum acceptable range of low ATP. But AMP will trigger AMP kinase, which is another enzyme, and that is the enzyme that is, mainly stimulated through low oxygen. So, low oxygen perfusion, hypoxia or hypoxemia. So that ultimately leads to hypoxia. Low energy supply for some other reason. So poisoning could cause this to occur. And also really, really high energy demand. So if demand outpaces supply because you're in high intensity training or something like that, then you'll get a buildup of AMP as well. So then you'll have GLUT4 translocate to the plasma membrane as well. So yes, GLUT4 is the primary way glucose is, in general conditions, entering cells in postprandial states for the average person. And also, yes, in a carnivore diet fed state, if you have even slight increases in the I/G ratio, you'll get some translocation of GLUT4 to the plasma membrane. However, typically you're in a fasted state, especially if you're carnivore. Typically you're going to be in a natural fasted state. In those situations, most glucose that's being taken up by your muscles and your brain, those are going to be taken up, particularly, I know for a fact, in muscles, skeletal muscle particularly, it's going to be through GLUT1. So if you actually observe people, most glucose uptake in muscles is going through GLUT1 as opposed to GLUT4. In the brain,
even with like high carb diets and insulin floating around, and
yeah, I actually believe even in people that are in eating high carb diets, the majority of glucose for use is going through GLUT1, at least whenever they're studied in a fasted state. So that does matter because, of course, it's not in the fed state. But GLUT1 is going to be depended on quite a bit. The reason that GLUT4 has so much emphasis placed on it is because it's insulin dependent. And I already explained why. So it's considered insulin dependent because you need insulin to even stimulate it to uptake glucose in the first place. All the others, GLUT1 through 3, that are involved in glucose metabolism, they are tightly integrated in the cell membrane. Like they don't sort of withdraw and come back into the cell into the cell cytosol.
So I suppose it almost sounds like GLUT4 is there to deal with, let's say, an emergency situation when there's too much glucose in the blood and it can be toxic and you need to get it out of there. And
yeah, I mean, basically, because I mean, you got to think GLUT4 in terms of quantity, like how much you actually have in each cell, is also dependent on the concentration of insulin. So you will have less GLUT4 whenever you are exhibiting less spikes and less absolute levels of insulin. So smaller area under the curve of insulin in a 24/7 period on a continuous basis. And that's due to insulin having those transcription factors on nuclear DNA to cause the genes to change in such a way that says, okay, well, hey, we need more GLUT4. We need more hexokinases involved in that pathway. We need more glucose metabolism enzymes and transporters. Like that's really what it involves. And so when you have less insulin, you're going to have less GLUT4. Like that's just a fact. And I think it's one of the reasons why you can have a manifestation, a presentation of peripheral insulin resistance when you don't have carbohydrates for long periods of time. There's this phenomenon that's been observed in people that have at least purported or reported to be consuming no carbohydrates, like for extended periods of time. And then when they have a glucose bolus and they do an oral glucose tolerance test, what they'll find is that their glucose spikes, which is normal given the circumstances. They consume carbohydrates. And then what happens is you would expect, according to the oral glucose tolerance test, if the longer it takes for the level to go back down to normal, to baseline, the less optimal your body is. Like that's really that's the idea. But it completely fails to account for the phenotypic expression of your genes. So if you're not consuming carbohydrates regularly and then you spike it, in these people that have been studied, it can hold before it comes back down. And they call that peripheral insulin resistance because despite the insulin concentration, it's not putting glucose back into the cells. At least immediately, as fast as they would like it to. And I have suggested, well, the only reason that insulin gets glucose down pretty fast is because you've got GLUT4 transporters that it translocates to the plasma membrane. But what if you don't have them? At least as many. Well, it's going to take a minute to produce them before it can get back down. It's not to say that the glucose can't go anywhere. I mean, obviously it does. You've got glucose can go through the other glucose-dependent transporters like GLUT1, GLUT2, GLUT3, and some of it can be used to synthesize glycogen, so you can fill your glycogen stores in your liver and your muscle cells. But you have a finite amount of those. I know that a lot of people online will try and act like you don't, and say, oh, well, glucose, it just fills your glycogen stores. It's like, until it doesn't. Then where does it go? But yeah, I think that you're right. Not to mention that insulin only really spikes. I mean, insulin will approach its final increase level. It approaches its final highest amount that it can go that's been observed. So it just starts to taper off and go what is considered zero order kinetics. It approaches that level right at about 130 milligrams per deciliter, actually. In the people studied, plus or minus a bit. But the question then becomes, well, after you eat a standard meal, what level does your glucose go to? Well, it goes beyond 130 milligrams per deciliter in many cases. Like it can go up to 180, 190. And in those circumstances, well, you're definitely sustaining lots of glycation damage. You really are, because insulin can only go up so much until eventually, then you start urinating glucose out. 180 milligrams per deciliter is the set point for that happening. You start urinating glucose out, which damages the kidneys, by the way. Mm-hmm. So, yeah.
So something else just came to me just then. I've heard Anthony Chaffee talk about this a couple of times. You mentioned you've had a chat to him, and he's suggesting that the insulin that we produce, that's produced in our bodies, in the pancreas, is not there primarily to deal with a glucose. It's there to deal with protein. Mm-hmm. And he sort of, I suppose, justifies that or bases that on the fact that diabetics who inject glucose, who inject insulin, they've now developed a fast-acting insulin because that's better at dealing with glucose spikes, and that type 2 diabetics who get rid of their type 2 diabetes by cutting out can move away from type 1 diabetics who stop eating. Sorry, let me get my thoughts straight. Yeah, type 1 diabetics who stop eating carbohydrates again can move away from injecting themselves with that fast-acting insulin and go back to the normal insulin that's basically replicates the one that the pancreas produces. Mm-hmm. So do you think there's any substance to that?
I'd have to actually look into that claim, because I only heard Anthony start making that claim less than a month ago.
Yeah.
I hadn't
2 videos within the last month or so. Yeah,
I hadn't heard that before. Because from my understanding, insulin was just one molecule. Like it is a specific hormone. It has a specific structure. But it wouldn't surprise me if they had to make a new one in order to get glucose down more readily, because the levels that these diabetics are exhibiting are far beyond, as I just stated, like insulin from our pancreas, it starts to taper off at 130 milligrams per deciliter, mm-hmm. like the levels at which it's secreted. So that doesn't necessarily give us an idea of the differing structures of insulin, so the different efficacy per molar unit of insulin between these two that he's purporting to be two different types. But what it does suggest is that if you're exhibiting levels higher than 130 milligrams per deciliter, you at the very least need to inject more insulin. So beyond the fact that you need to inject insulin tells you something. Like that in and of itself is an inference. Like, okay, well, should we be spiking our sugar all the time? Like, if ever, really, especially mm-hmm. in our modern climate, it's like, well, no, not at all. But yeah, that's an interesting. It's an interesting position and claim. I really would have to look into it, and I'd also have to look into the mechanisms, because also the prevailing idea, again, from my understanding, science is continuously changing, is that the reason for sort of insulin resistance as proxy measure, not directly measured, because you can't directly measure it, but as proxy measured, the idea behind why that starts to occur and why fasting insulin tends to creep upward more and more and more is because the more fat you store, the more hypertrophied your fat cells and your muscle cells are. And every single cell has a maximum rate of hypertrophy, but also has a maximum rate of hyperplasia, the rate at which the body can actually divide to create more and more cells in the same tissue, right? And after you reach your genotypic maximum rate of hyperplasia, then you start to fill all of your cells up more and more until you reach your maximum rate of hypertrophy. And then, well, as that's continuing to go up, you're approaching your maximum rate of hypertrophy and hyperplasia, the insulin starts to creep up too, because now there needs to be more insulin present to get the same level of glucose down. Like that's the idea behind insulin resistance in the quote unquote pathological sense, as opposed to the peripheral sense, like I was explaining earlier. And so the reason I bring that up is because if Anthony is saying that they've created a new insulin, and again, I'm not saying I don't believe this, it's just I'm thinking aloud here. If they've created a new insulin that is purported to be more effective at bringing down glucose levels than previous insulin, then it doesn't it would kind of shoot that theory of insulin resistance in the foot, would it not? Because, I mean, how can you speed up the rate of the glucose, what do they call it? Glucose clearance, if the rate of glucose clearance is slowed because of a buildup of fat, like you're plugged up, so to speak, your system is plugged up. And so I'd be interested in understanding, really what I'm curious to see is I don't disbelieve that there is a new type of insulin that's being created. Like, it seems very plausible, and I can look into it myself. But I'm more curious about what the mechanism is how it even does that.
Okay. Yeah.
How it's more effective. Yeah.
Why they call it fast acting and why it...
Yeah. Yeah, exactly. I wish I had an answer to that, because that is fascinating. And yeah, I've heard him make it I've heard the claim about two times from him. So.
Okay. Yeah, I suppose that was a long discussion on what I thought was going to be a simple question, which is great because I love delving into things like that.
Yeah, good. Yeah.
So I suppose going on that gluconeogenesis, I've got a few questions on that, and also the Randall cycle. I suppose let's do the Randall cycle one first. Is the Randall cycle just sort of like, this sort of fader switch between glucose use and fat use, or do things like ketones and play a factor in? I think I remember Sarah Pugh. I don't know if you watch any. She mentioned once as a passing comment in a video, and it sort of stuck with me, maybe a year ago, that the ketones do play a factor in the Randall cycle, but I haven't got time to talk about that now. And it just stuck with me, and I'm trying to sort of see if there's an answer to that. Do ketones come into it at all?
Yeah, and it's complicated, but it's also very simple at the same time. It's very, very weird. So the Randall cycle itself is very poorly named. It's a shame that it is named a cycle in the first place, because everyone thinks, when you think about a Krebs cycle, for example, the Krebs cycle, it's a cycle. It's literally a cycle. You start with one intermediate, and then 8 steps go around, and it comes right back around to the same molecule. That's like, and then eventually it fades out, fizzles out, because you can't recycle everything forever, and then that's where anaplerosis comes in. But it's a cycle. The Randall cycle is a cross inhibition of 2 pathways. And now I wrote about in my book why it was called a cycle, and I really tried to steelman the case for calling it a cycle, even though I disagreed with it. And still, even then, it was really difficult to do so. I really don't know what Randall was thinking. I looked into his papers, like I read the very first paper that he wrote about it, and it just didn't make any sense. It didn't make any sense to me. Now, granted, now that there's been future research done, there was particularly a paper written in 2012 about this. It was like the Randall cycle revisited, and it was just an aggregation of a ton of research done on this topic into one paper. That wasn't a good paper either for other reasons. However, it was sufficient enough to get a good idea of what was going on. And that's where those graphics, those infamous graphics come in, where you've got the two, the Randall, the glucose, glycolysis inhibiting fatty acid oxidation, and then you've got the opposite too, just depending on circumstance. Now, The main catalyst for the Randle cycle itself is maximum rate of respiration of mitochondria. It's a buildup of ATP. And what happens when you get a buildup of ATP, since your cells, they can only deal with so much. If a system gets too much energy, there's always a problem that happens in whether it's a human manufactured system or it's a biological system. There's always ways to mitigate that, or else something will go. You can't have too much energy in the system. That's a problem. And so what happens is ATP will inhibit certain elements of the Krebs cycle, which will cause a molecule of isocitrate to build up. Well, isocitrate can't move forward in the reaction now because you've got too much ATP. So it backflows, it reverses, since it's a reversible reaction that creates isocitrate back to citrate. Citrate can't backflow back to acetyl-CoA and oxaloacetate. Can't do any of that. So citrate now builds up in the mitochondria. Well, citrate has an ability to migrate from the mitochondria into the cytosol, the cell fluid, and that's exactly what happens. So citrate will move out of the fluid. Now, depending on primarily the I to G ratio, insulin to glucagon ratio, citrate will either exist as citrate, or it will actually go through a further step where it produces acetyl-CoA and then malonyl-CoA. So if you refer to these two graphics that I've, if you've watched the Randle cycle module that I've done, there's one that it shows malonyl-CoA having been created, which will inhibit fatty acid oxidation. That's the molecule that inhibits CPT1 on the mitochondria. That's what inhibits the pathway. That's what causes fats to pool in the cell, which will ultimately be used to create triglycerides, the storage form of fat. So that's why, if you're consuming a mixed meal and you've got a high I to G ratio, there's a large influence on all the fat that you're consuming to be created into triglycerides. I think this is the primary reason why the myth came to be that fat makes you fat. Because, well, first of all, super easy to think about that because it's like, well, I mean, this is on my body. So if I just remove this, then I'll get rid of it. But the problem is that the results that occurred after people abstained from fat didn't help them. It didn't help with disabusing them of that notion, because you actually do see fat loss if you cut out all your fat and you replace it with plants. So it didn't help that it actually did happen to be the case. And it does happen to be the case that if you go vegan or something, you'll lose lots of fat. So it doesn't help the case. But that's just one part of the picture. Buildup of citrate. Insulin causes citrate. It has a large influence on activating a certain enzyme to produce malonyl-CoA. I think it's fatty acid synthase actually is a big part of that. It could be wrong. Actually, I believe that's afterwards. I can't remember the specific enzyme involved in citrate's conversion to malonyl-CoA. But I think it's acetyl-CoA carboxylase actually. But again, anyone watching, correct me if I'm wrong. But that's just one part of the picture though, because Ben Bickman did a video that I reacted to a while back on the Randle cycle, and it's not like, obviously since I'm like doing a video on him, I'm being like really like right. But in reality, the video wasn't the worst video in the world. My biggest problem was that he was saying that insulin's a dictator of fuel use, when in reality, if that were the case, insulin resistance wouldn't exist. Like, that's not, you can't have insulin resistance if insulin's a dictator of fuel use. I think he brought a very good point to the discussion, which is that insulin itself it's completely, the graphics themselves they don't consider insulin and glucagon at all. Like, that's like a broad overarching aspect of this whole system, because citrate outside of the context of a high I to G ratio will not convert into malonyl-CoA. Instead, it will inhibit glycolysis. Citrate is what inhibits glycolysis. GLUT4 directly. Again, in the graphics, it makes it look like GLUT4 is like integrated into the cell membrane. That's not true. If it's inhibited, it means it's not being translocated to the plasma membrane. GLUT4 is inhibited. PFK1, PFK2 is inhibited. And then you've also got other substrates like long-chain fatty acyl-CoA molecules that directly inhibit pyruvate kinase. You've got acetyl-CoA inhibiting in the mitochondria PDH, pyruvate dehydrogenase, as well. So it depends on insulin, depends on glucagon, but it also depends on the amount of GLUT4 you have. It also depends on the inertia of the mitochondria. So what are they using at that time? It seems to be a big aspect of it. Those two go hand in hand though. And so the reason why I'm bringing all this up, I just want to explain that first, because ultimately the thing that causes the cross inhibition, the thing that causes an inhibition of either pathway, is a buildup of ATP. So when Sarah Pugh says something like ketones play a role in the Randall cycle, the reason she says that is because ketones play a role in the production of ATP. So ketones they don't go through fatty acid oxidation, they don't go through beta oxidation, and they don't go through glycolysis. They actually go through they permeate very easily. There's not even a set like pathway that it goes through. They enter the mitochondria like directly. They can pass through a cell. They go right through into the mitochondria. There are transporters that it goes through, but it's passive. It's not facilitated. And they then go through a very, very short process of ketolysis. They're very, very quickly converted into acetyl-CoA. And acetyl-CoA is the first intermediate, really, well, I would say it's really the first intermediate of the Krebs cycle. I know that people say citrate, which is a technicality as opposed to a practicality, because really it's acetyl-CoA forming citrate with oxaloacetate. So acetyl-CoA it helps to form the first step of ATP production. And so it does influence Randall cycle activity because it's involved in ATP production. But it's not involved in the cross inhibition of the two pathways. That's not true.
Okay.
Because the cross inhibition is strictly to do with fatty acids and glucose. So
okay. Yeah. Okay.
Yeah.
That makes sense.
Okay.
Just on ketones, a couple of questions there.
Okay.
Is everyone producing ketones all the time? So even people on a low-fat, high-carb diet, would they be producing ketones?
Yes.
Okay. So
yes,
the way
it's a small amount.
Understood it is when you have a high-fat diet, the excess fat gets converted to ketones. But that's not exactly right.
Yeah, I mean, yeah, there's some nuance there, because even in people that are studied on a standard American diet, even people that are like in a fed state, there is a small amount of ketone. There are small amounts of ketones that are produced. That in and of itself is actually a very strong inference that we are not designed to eat lots of carbohydrates. And the reason for that is because in order to have a certain level of ketone production, you have to have a certain level of fatty acid oxidation, and it's got to be to a degree that your liver sort of quote unquote can't keep up with in terms of it producing energy from it. Because the reason that you produce ketones actually is because this will get a little complicated. So apologies in advance if it gets a little too convoluted. But the reason that ketones are produced is because when your liver is undergoing lots of fat oxidation, it's going through beta oxidation, it's producing energy like normal, like everything's going as usual. But what you're also doing typically when you're undergoing lots of fat oxidation is you're producing lots of glucose because you're not consuming it. So it's also happening in the same type of cell, like a liver cell, a hepatocyte. Well, the first intermediate of producing, the first step in producing ATP from fat oxidation really is acetyl-CoA joining with a molecule of oxaloacetate in the Krebs cycle. So OAA is usually how that's abbreviated. Well, oxaloacetate also happens to be the first intermediate, the most common one used to create glucose in the liver. So basically you can think of oxaloacetate as like it's a point on a graph, and you've got two lines coming from that point. You've got fatty acid oxidation in one of them, and you've got gluconeogenesis as another. And so basically these two pathways are drawing from the same pool. Okay, so what happens is the concentration starts to decrease. The concentration of oxaloacetate starts to decrease because you've got two pathways that are drawing from this intermediate, this substrate. And so as a result of that, less acetyl-CoA produced from fat can actually join with oxaloacetate to produce ATP. So what happens is that the rate of ATP production, it stays relatively the same, but the amount of acetyl-CoA that's being produced, it needs to be shuttled towards a different pathway. And so these molecules of acetyl-CoA start getting stacked on top of each other, and they start to effectively, and that's what produces ketone bodies. You've got beta-hydroxybutyrate, and you've got acetoacetate. Then they leak, sounds like it's like unintentional and not what the body's designed to do, but let's just use the word anyway, noting that that's not exactly true. They leak out from the liver, So that the liver, they're administered into the bloodstream is probably a better way of putting it. And then they're picked up by any tissues that are in need of ATP at that time. So they can kind of like, if a cell is oxidizing fat, ketones can kind of just squeeze in once in a while and just be like, hey, I'm here. So I'm going to come in and I'll be used to create ATP as well. It's important to note that ketones are not subsisted on primarily by any cell except neurons in a ketogenic state. And that's only because fatty acids can't pass through the blood-brain barrier, except for like an extremely small amount of them. They happen to be the polyunsaturated fats, which is no coincidence. It's because they're designed for the brain structure. So
yeah. Not to produce ATP.
Yeah, exactly. But like your muscle cells, your heart, your cardiac cells, so still muscle, but just different type. They do not subsist primarily on ketones. Like your cells in a ketogenic state are primarily using fatty acids. But this is why ketones are produced. And of course, people have used this to suggest that ketosis is like a bad thing, but that's going to get to my point. Because what they say is, oh, well, that's a sign that you're overburdening your liver with fat. Well, if that's the case, then why is it that in a high carbohydrate fed state, you still produce ketones?
Okay, yeah.
Of course, they could say, well, it's because those people are also overburdening their liver with fat because they're eating a high fat standard American diet, right? But it doesn't matter what diet you're eating. You're always producing ketones. It doesn't matter if you cut out fat.
Even a low fat, high carb diet will produce ketones.
Yes. It has been observed in these people. Now, granted, since this is a more fringe, nuanced discussion and topic, there's probably a lot less observations than many other observations. And are there specific studies that are designed to look at that in a high carb, low fat population? Maybe not. I haven't looked hard enough, but I haven't looked at all actually. But of the ones that I have seen encountered, yes, that they do produce ketones, and that raises a question though. Then, I mean, if you're going to say that we're not designed to be a fat burning quote unquote species, we're not supposed to eat high fat, we're not supposed to eat low carb or no carb. Well, why is it that your liver insists, you can think of it as insisting, that it produces some ketones? It insists on oxidizing fat. It insists on oxidizing fat, at least to some extent, because that's the only way that you're going to be producing ketones. Like that's the, according to that model, that's the only way that you're going to be doing it. And so is it the strongest inference? No. That's why I don't often talk about it. I think there's a lot stronger inferences that usually pertain to anatomy. But it's yes, it is true that most people, everyone is producing ketones to some degree. It's just higher or lower depending on what you're doing.
Okay. Okay. Yep. That makes sense.
Okay. Good.
Sorry, let me just have a look at my notes quickly.
Yeah, that's fine.
So I suppose just more of a broad question. When people say someone's in ketosis, what does that actually mean?
It's a good question. Usually it's based off of an arbitrary cutoff of a ketone reading, which is very flawed because you can have adjustments in your, you can have variations in your ketone reading and ketone meter, depending on how fat adapted you are. I think that's the main problem. Ketosis typically, colloquially, means that you are in a state of primarily fatty acid oxidation and ketone oxidation as well, with very minimal carbohydrate oxidation, or really pyruvate oxidation. I'm a stickler for that language, but I think the best way of actually assessing whether someone's in ketosis then is not by looking at a ketone meter, because that's really, that gets to the heart of your question. It's like, how do I know if I'm in ketosis? Like, okay, if that's what ketosis colloquially means, and we're going to work with that functional definition, how do I actually determine, like, how do we know when someone's in ketosis then? Most people will say, look at a ketone meter, but that's not reliable, because some people on a carnivore diet can actually see that despite them being fat adapted, because they're not eating carbohydrates and they're not really overeating on protein and fat as much as they might be led to believe this because of what they're seeing is that they are actually having lower and lower ketone readings. And I've said, well, hold on a minute. I actually made a video specifically about this.
Yeah,
you may have seen it.
Yeah.
Yeah, where it's like, well, hold on a second. Can your rate of ketone production go down if you are becoming more efficient at oxidizing fat? Well, theoretically, yeah, because of the mechanism that I just explained. Okay. even if that weren't true, do we know that people become more efficient at oxidizing a certain substrate the more that they do it? So ketones, when they are produced, well,
So
yeah,
you're using more of them, so there's less floating around to be measured.
right, exactly. And so what I did was I was like, well, I I don't believe that the ketometer is the strongest inference. There seems to be limitations with that. So what is the strongest inference? What do we do to determine whether someone's in ketosis or not? Well, you would look at what they're actually oxidizing for fuel, and we actually know how to do this, which is with a respiratory exchange ratio, or really it's also known as RQ, respiratory quotient, which is more accurate because it is a quotient, not a ratio. But they go hand in hand, I understand, but still. And that actually measures the ratio of carbon dioxide to oxygen, which are, oxygen comes in, carbon dioxide comes out. You can actually measure what's on a system-wide level. So not on a cellular level, but we're looking at the system-wide level. That's what we're really trying to assess, what the body's primarily oxidizing. So a quotient of 0.7 is like pure fatty acid oxidation. And it's between 0.7 and 0.9, you're in ketosis, basically. And then once you get to about a quotient of 1, like exactly 1.00, maybe slightly more, then you're basically undergoing pure carbohydrate oxidation. So usually this happens in PhD settings, or like in laboratory settings with PhDs, where they hook people up with a respiratory gas analysis device and they tell them to exercise. And what they'll actually measure is a gradual increase in the respiratory quotient over time, because your body shifts over to carbohydrate metabolism almost strictly during exercise. And it's because of the enormous amount of pyruvate that's produced during exercise whenever you're employing lots of exercise, muscle contractions. So, yeah.
Yeah, I was actually fortunate enough to have that done, not for that purpose, just to as a VO2 max test.
Yeah, they do that too. Yeah.
When I was learning to be a fitness instructor, the guy that was teaching us was, a physiology teacher from a university. So he took us into his lab, and I got to put the respirator on and start running and then sprinting on a treadmill, and got my VO2 max tested. And yeah, it was very interesting.
Yeah.
They weren't talking about it. I mean, we weren't talking about it back then from a glucose versus fat metabolism. It was just to get your VO2 max done.
Mm-hmm. Yeah, those things are expensive. They're very expensive.
Yeah. So I was very fortunate to,
yeah.
to have that.
Yeah.
Okay. I think that was everything that I had on ketosis. I suppose leading on from that, and sort of going back to something that you said just before, and you sort of say frequently, just, what makes you and me and us believe that we should be primarily oxidizing fat as opposed to carbohydrates. So most mammals, as far as I understand it, even like, let's say, a cow who's a pure vegan, they don't consume carbohydrates as such. Like they eat the grass, and the grass is converted, for lack of a better word, into fats and proteins. So they would be in ketosis as well, I guess.
You got it.
Is that right?
Yep, exactly. As one of the main things I touch on now as well, is like the idea that we should be consuming simple carbohydrates in large quantities regularly, multiple times a day, is asinine on its face. Because if you look at it from an evolutionary point of view, no terrestrial animal, no aquatic animal encounters significant amounts of simple carbohydrates. Those are ephemeral and transient. Those are only acquired during fruit season. Now, fruit season untouched by human intervention
is a few weeks a
year. is a few weeks a year, and also involves fruit that still doesn't have too many simple carbs in it. It's primarily, it's starchy, or really it's not starchy at all. It's fibrous is what I meant to say. Not starchy, it's fibrous. And so, yeah, it does have simple carbs in there, sure, yeah. It has some sugar alcohols and all that. But definitely, like, you're not no animal was consuming that. Now, not to mention, for example, like cats or lions, they don't even have sweet receptors. So they're not eating it at all. There's not even a desire for them to eat that stuff. They don't have that desire because it's not even sweet to them. So that's also something to consider as well. But the other thing is, put aside simple carbs, also starches. Starches are even less ancestrally consistent because now you're talking about things that we never, like, we encountered it even less than fruit, because fruit, it has fiber and it has simple sugars, simple carbs. Starches, I mean, there were some like ever so slight amounts of starch that we would have encountered, like but like pulling roots from the ground.
Yeah,
it's
the tubers that we used to sort of.
Yes, like tubers. Yeah, exactly. But we've mainly increased the starch quantity in very recent times, like massively. And so I feel like we've been consuming starch for even less amounts of time than we've been consuming simple carbs. Because I mean, we really started consuming starch, post agriculture. Simple carbs, we increased the frequency post agriculture, and then ultimately the quantity, as we know. But it took a while because we still had to graft and hybridize the plants, which takes some time. So yeah, but the main thing that I say is like, okay, well, you're saying oxidizing fat's a bad thing. Oxidizing ketones is a bad thing. We should be oxidizing pyruvate mainly. We should be consuming lots of glucose. It's like, well, literally no animal on the face of the planet does that.
Yeah.
Except for like sunbirds and hummingbirds. Yes.
Because the arguments that, when I've had discussions with a couple of vegan friends, is like, oh, don't you want to be like a gorilla or a chimpanzee? Look at them, they're jacked and everything. They said, and my counter to that was, yeah, but okay, let's look at a gorilla. They sit around and chew leaves for 6 or 7 or 8 hours a day, and in the end, they produce fats and proteins, and that's what they actually.
Yeah, they have a large cecum that is able to metabolize, or not metabolize, but break those compounds down into their vitamins, their minerals, their amino acids, their fats. That's why they look that way. Not to mention that even then they're still having to eat regularly. They're still having to eat regularly to do that. So that's why, like, we don't have the ability to ferment fiber into all of those compounds. Like we have a very small capacity to do that. Yes, we do produce butyrate. Yes, in small quantities. Usually it's isolated to a very focal part of the small intestine, its usage. We do produce propionate. We do produce, I think it might be acetate is the other one. And then you do produce also aldehydes and alcohol, by the way, but whatever.
Is that, sorry, that's the soluble fiber that we can do that with, or is it the insoluble?
No, it's the soluble.
Soluble.
It's the soluble. Yeah, it's also
the insoluble. It just passes through and destroys your gut.
Yes. Yes. Exactly. Yeah, the soluble fiber. It also is the one that creates the gel-like structure in the digestive system, because the gel. Well, I mean, look, the gel is not only taking up. Everyone will say, "Well, it sops up toxins," and it's like, yeah, it's also sopping up all of your, not all of them, but much of your nutrition. Actually, like, it is an anti-nutrient by definition. Soluble fiber, it is. So, if people want to make the argument that, oh, well, the amount of nutrition that's lost is what we're designed to lose in the natural process because we're designed to eat fiber, it's like, well, then why can't we digest the thing? Like, we have a very small capacity. We have the same capacity to work with fiber digestively as like a cat does. And you wouldn't say cats are designed to eat fiber. So
yeah, exactly.
Yeah.
Sorry. Got a house full of kids, and I told them to stay out of here, but they're trying to come in.
That's okay.
Yeah, and I suppose my other argument is, in that respect, was like, and then what you also have to do is turn around and eat your poo to get your.
Yeah, exactly. To get B12 and B12. Like all that stuff. Yes.
So go ahead and be like a gorilla if you want.
Yeah, go ahead. Yeah.
Just a side question on that. I suppose it comes back to muscle building. We need to sort of do resistance training to build big muscles. Gorillas and chimpanzees, and how do they get so muscly, if you like?
Yeah,
They
well,
don't sit there and do bench press and squats. And
right. Yeah. Yeah, I mean it's an interesting question because also, I mean before I even directly answer it, what I will say is that the physiques that we see nowadays that are more regularly encountered, well, at least encountered online, a lot of them they do use steroids to get to that degree. So we also have to eliminate those from the pool to get a more objective view of like the genotypic set point for muscle on human beings. If you look at like other African tribes, they're more robust in height. But and also muscle, but you'll also see that they're still actually quite skinny. Like they're not like ripped, they're not like a human
yes.
version of gorillas. And
Hmm.
so I don't think that we're designed to at least get to the gorilla point. But the question still remains: is how do gorillas get to that point then? Like how do they get to that point?
Yeah.
And that's why I say it's an interesting question because I mean, yes, gorillas, for example, they're not like other monkeys where they're like climbing on everything all the time, you know. And by the way, not to mention, even if they were, that would also leave a question as to why the monkeys that are climbing on everything all the time aren't ripped themselves because they're not. They're not
Yeah,
like they don't look like that.
the smaller ones, yeah.
Yeah,
Chimpanzees, I think, are quite
yeah,
built.
yeah, yeah, yeah.
Most
Yeah, I had a
most
picture in my head of
monkeys
the
are.
smaller ones. Yeah,
Yeah, yeah,
and I think that just has to do with the increased body weight. Like if they're hanging around, like if they have more weight to carry around when they're swinging around, I mean, that's just going to make sense. They have more muscle to you know effectuate that movement. For gorillas, I'm not even. I don't really know. I don't know. I mean, I know that for example, there are sculptures of human beings that were made during like the Greek times, and they had
yeah.
like yeah. And you look at those people, and you're like, how did they get to that point? If they weren't like, were they engaging in lots of resistance training, like just not in like a barbell form, but with all the other
Yeah,
stuff?
but
Well, I
in
mean, yeah, in many cases, like a lot of them were, but even then, that was like that the statues were built that way because that was like the ideal. That was an outlier, but it still remains like okay, they couldn't have sculpted that without having seen it. So it's clearly we had attained it back then.
yeah, I think this was a conversation you had with Dr. John.
Jakewish, yes.
Yeah,
Yeah. Okay, yeah. So you did see that, yeah, because we kind of arrived. I wanted to ask him that. I was like, yeah. So what do you have to say about it? I wasn't even really looking for like a guided answer. Like I was, it was just for the audience. I was like, yeah, like, hey, just out of curiosity, what do you think about the about that? Because I think that it might have been sort of anomalous, like a genetic anomaly. Now I know that might seem like a unsatisfying sort of scapegoat or a convenient answer, but I mean, just because it does sound like that doesn't mean that that's why I'm making that conclusion. Because we do see that this can happen in other animals as well. Like a cow can have a myostatin dysregulation, and they can actually have extreme. They get so muscular that it actually harms them. I don't know if you've ever seen cows like that before, uh, but
there's a breed of cows that are huge. Is that the ones you're talking about? Is that something different?
well, they could have something to do, that could be part of it. Cows that have myostatin problems, but there are certain cows that they were genetically modified, and it was they're not like regularly seen. Like they actually, it's like comically large. Their muscles are like they're swallowing them, and
Yeah. Oh, yeah, I actually have seen a picture. Yeah,
yeah, and those cows. Well, I mean, they weren't working out at all.
yeah. Other than walking around carrying the weight, but is that
Right.
enough to make them that big?
Exactly. And so it's not to say that gorillas are like genetically modified or anything like that. But what that does suggest is that you don't have to necessarily work out and engage in resistance training to get muscle on your body. So they might have different genetic variations that influence, they definitely do, that influence resting muscle outside of the context of as much resistance training as we would need to employ in order to get to similar high muscle levels or mass on our body. So I know that might not be the most satisfying answer, even for me. But I mean,
No, and I sort of put you on the spot with that. I didn't expect you to understand, gorilla metabolism and gorilla.
no, it's fine. It's fine. Like I like those
But,
questions.
yeah, it's
Yeah.
sort of. I mean, given that we're supposedly so closely related, like, yeah, why did they get so big and jacked? And they
Well,
don't
yeah, and I
go to
also
a
want to
gym.
say a lot of people will say, especially the vegans, will say they're our closest ancestors. When in reality, they're not our closest ancestors. It's not. It's that they're our least distant.
Yes, yeah.
because they're still pretty distant ancestors. Yeah.
And they're not that far off from us being related to a banana, I think, genetically
Yeah.
speaking.
Yeah. Like, yeah, exactly.
Just on, I mean, touched on Dr. John Jaquish. Am I saying his name correctly? Um,
Yeah.
so I did have a question from that later on, but since we talked about it, I've heard him speak about hyperplasia
Yeah.
versus hypertrophy. And from what I recall, he seems to think, and I was always taught that you can't get hyperplasia in muscles in a human, but he seems to suggest that it is possible. Have you looked into that at all?
Yeah. A bit. And it's extremely small amounts that you can get
And I
in
think
human beings.
his regimen for doing it is obviously working out hard with an X3 bar, but then
Glycogen replenishment and all that stuff.
yeah, stretching like
Yes.
stretching
Stretching.
immediately afterwards, and then 20 or 30 grams of glycogen within 30 minutes of the workout, and you can start having some hyperplasia.
Yeah, I think that it's an extremely small amount, if any at all, in the studies that he was referencing.
Okay.
And I don't mean any disrespect to Dr. Jaquish for it, for obvious reasons. But hyperplasia, yeah, usually you were born. A lot of people actually they know this, but they also don't put two and two together because it's actually pretty interesting that the muscle cells that you're born with, you're pretty much not going to have any more or less than that. Like that's what you have.
And
Um,
that's what they mean when someone's got good genetics, I suppose.
Yeah, well, I mean that helps. Like it does. If you've got more muscle or more muscle fibers to build, like yeah, that's going to help with your strength, your resting strength, and also the strength, your genotypic maximum that you can get to. Um, but yeah, I remember reading that, and I'm not really convinced. I'm not really convinced. That was probably the only part of his book that I was really like, eh, I don't know about that one. I mean, obviously, there's some other ways in which he phrased things that like using the word risk and stuff. Like, obviously, I'm
Yeah.
like, but it was one of those things where it's like everyone uses that. So I'm not going to say that it exonerates him, but it's like, okay, I can kind of just brush past. I'm so used to this. Like, it didn't stand out. The thing that stood out the most was that claim. I'm not convinced, but um, perhaps I'm wrong. Who knows? We do absolutely know that you're not going to get a ton in human beings. That's not going to happen. Like you're not. Cats can do it,
Okay.
but
They can increase number of
yeah, yeah,
muscle
yeah.
fibers they have. Okay.
Cats can. Yep, not humans.
Okay. And as yeah, it sort of goes back to building muscle to some extent is good, but beyond that is probably not good for us. You know,
Yeah,
getting into
generally.
the bodybuilder type territory,
Yeah, yeah, yeah. It puts a strain on
and
the
and
heart
that
eventually,
and that
and
might be one of the justifications for having that sort of thought as well. Like, yeah, if we were meant to get that big, we will probably be able to increase the number of muscle fibers.
yeah, maybe. Yeah, yeah. So it's either we didn't develop a positive selection pressure for that because it wasn't necessary, or there was actually maybe we did have that ability theoretically, and there was a negative selection pressure against that because it interfered with the survival of the organism to reproductive age because it was bad for us or something.
Yeah.
That's how the theory goes. So anyway,
Okay. Um. Sorry. How are we going for time? Like, how much time do you have?
well, if you, I mean, pretty much as long as you need to get the questions out of the way. Um,
I could keep going for another couple of hours. So
well, okay, so let's do. I've got
cut me off when.
yeah. Let's do 45 minutes. I have 45 more
Okay.
minutes.
Okay. So I'm going to sort of jump ahead um, with some of the questions, but sort of going back to since we're talking about muscles, can I throw some muscle questions at you?
Yes, that's one of my
Okay.
favorite topics.
Um, the actin myosin cross bridge,
Yep.
it's still referred to as a theory. Is it a theory or does it actually occur, if you like?
From my understanding, it's not really a theory. It's actually been observed.
Yeah,
But I
that's
mean,
that's what I thought too. But when I was taught it, and even now when people are talking about it, they're calling it a
I
theory.
think it's not that the actin filaments and the myosin chains, they do exist. I think if there's going to be any theory, it's the way in which they bind and then shorten. That's how but in reality, the theory has withstood decades of observations. And so it's not to say that it's necessarily like unequivocally true per se, but from my understanding, that's what's been observed, is that there have been actual contraction, a shortening of. We know that muscle fibers shorten during a contraction. Myosin heads exist, myosin chains, whatever you want to call them. Actin filaments exist because that's part of the cytoskeleton. They're part of forming a constriction ring in mitosis. Like we know all this. So the only theory that there is that the thing that causes the muscle fiber to shorten, I suppose the only theory would be, is the binding of myosin to actin, and then a sort of rowboat motion that's formed or occurring that causes the fiber to actually shorten. But I mean, it's the theory that helps to explain it to the greatest degree possible. And look, I'm not foreign to the idea that just because there's a theory that explains things really well, that must mean that it's true. In fact, I know that what I'm saying is that it's not a foreign idea that just because a story makes sense, it means it's true, is false. I'm not foreign to, like that. That's absolutely, that's false. That is false. Just because a story makes sense doesn't mean that it's true, because I have to deal with this all the time when I talk about proton pumping. It's like proton pumping is not a thing. That's not possible.
Mm-hmm.
And I know that people will say, again, people's immediate response is like, well, it makes so much sense though. Like, obviously, it has to be. It's like, no, yeah, but no, it's not. And so could there be something more complicated going on? I suppose. And that's something that I should probably look into. But I haven't really looked. I haven't, what would you say? I've kind of just glossed over that detail because I'm like, well, the actual implications, like the necessary elements to understand in order to, for broad application of a muscle fiber and muscle energetics is quite insignificant by my estimation. But I am someone that likes exactitude, so I mean, I suppose it's something that I should look into. I didn't know it was ever even considered a theory.
Yeah, when I was taught it originally, it was called a theory, and even now, when you hear people talk about it, I think they still refer to it as the crossbridge, actin-myosin crossbridge theory.
It's interesting because I feel like we would have the technology to observe that.
Yeah, that's what I would have thought. Electron microscopes.
Yeah, exactly. So that's why I say maybe they just, it's like a vestige, a theory. Yeah. I'm not sure. That's something that I should look into as well. Yeah.
So delving a little bit deeper into how it all works, and then trying to remember from 20 years ago. So, as far as I can recall, what initiates the muscle contraction is a nerve impulse coming in, so calcium getting into the muscle cell, which then is it troponin that gets exposed, which is the myosin binding
site
on the actin, and the head of the myosin reaches up and then pulls across, and then it's got to sort of let go and reset. And the thing that I sort of found strange is how ATP comes into all of this. And then, correct me if I'm wrong, and I suppose this is my question, ATP is required to get the myosin to release from the actin.
To release, yes.
And flick back into its ready position. Is that correct?
Uh-huh.
So the actual contraction itself doesn't require ATP? It's only the contraction.
No, the contraction actually requires ATP, and also the ATP is required to get rid of to disconnect it. Yeah. It's both.
Okay. Oh, it is both. Okay.
Yeah.
Well, then where does ATP come into it in terms of the contraction side of things?
In terms of the specifics, so what you said was correct. Usually when the calcium comes in, it's referred to as polarization. When a muscle cell is polarized, right? And the reason I put it in quotes is because in reality, it's not polarization because of electroneutrality. So
Yes,
people
otherwise
forget.
it'd be a walking battery.
Yeah. I mean, it's like if the law of electroneutral weren't the case and weren't persisting at all times, then as soon as you consume salt, you would explode. Like or you consume like an ion or something, you would explode. Like, because even the smallest charge differentials release massive discharges of current. They produce massive discharges of currents. It has to do with Coulomb's force. So that's not, when they say polarizing and they say, oh, here's a voltage, because they take an electrode and they put it in there and say they read a voltage. It was produced by the electrode. It was not actually seen in the fluid pool, because that's not true. You just get a change in pH. So anyway, yes, you're right about that. In terms of the specific, like ATP coming in, and yes, it unlinks the myosin from the myosin heads or the myosin chains or whatever you want to call them, from the actin filaments or the microfilaments. There's that. And then we know that glycogen is required for every muscle twitch, because it produces a massive amount of ATP in a very short amount of time, beyond the rate at which the mitochondria could ever imagine producing ATP. Like, there's just a rate of respiration that cannot be attained at normal resting levels in mitochondria. And so I'll put that to the side for a minute. But in terms of the actual mechanism by which ATP is required, it's really just through hydrolysis. It's hydrolysis of ATP to release chemical potential energy. Now, where is that chemical potential energy being activated? Like, look, is there a specific site for it? There might be. I don't remember the absolute specifics. I was never taught that part. I just know that usually when the body requires ATP or requires energy in the form of ATP hydrolyzed, it's hydrolyzed, it forms chemical potential energy, and then it's introduced into a certain step of a pathway. Like, that's really, it's used. There's a lot of I guess you could say there's a lot of mystery surrounding energy in the first place, like even defining it, even, all that stuff. So I guess it's a little arcane. But just because I don't know it doesn't mean it's arcane. It could just be because I don't actually know the specific part. But it's through hydrolysis, absolutely. Like, you're going to get ATP and get ADP and PI at that site. So
Okay. That was one of my questions too. Energy, like, we throw around this word energy and people sort of throw it around, and I think for the most part have no real understanding of what energy is or what it's supposed to be. So when we say or that ATP is the energy currency of the body, does that mean that ATP is used to initiate other chemical reactions, and without ATP they wouldn't happen? Or how does that sort of...
Yeah, I mean, this also gets into a conversation about thermodynamics and a more fundamental conversation about physics and like Gibbs free energy, and also delta G, so the change in, Gibbs energy, and also a conversation about endergonic versus exergonic reactions as well.
Oh, okay.
Yeah. There's a lot to go on there as well. Yes, energy, chemical potential energy is required to effectuate certain biological processes, and without that chemical potential energy, they would not occur. That is true. There are certain reactions
is that what ATP does?
Yeah. So ATP is hydrolyzed, and it releases that chemical potential energy that is not really observed, because energy, it's not even something that can be directly measured and all this stuff. Yeah, you're getting really fundamental now, because that's something that plagues the minds of scientists and thinkers all across the world. Like, we don't even really know a lot of it. We refer to this inexplicable phenomenon as energy.
Mm-hmm. And
then we divvied up into different categories, like kinetic energy and then potential energy and all this stuff. And it's a construct. It's a construct. And I'm not saying that just because something's a construct, it means that it's bad. I mean, we need some sort of order in the world. We need to do what we can with the chaos around us. But with that said, that does mean that there are going to be some unanswered questions. But what we do know is that, you need energy is required for things to move against concentration gradients. That's obvious. So if something's going from low concentration to high concentration, it's going to require energy. That's when you get into the conversation about passive versus active transport. So active transport requires ATP, requires chemical potential energy, because it's moving from low concentration to high concentration. It involves moving a substrate from low concentration to high concentration. And then passive transport is the opposite. It doesn't require ATP at all, because, areas of high concentration tend to flow from to flow from to areas of low concentration. Or yes, sorry, I
Yes.
tripped myself up there. But what I was getting at was that there are other reactions that, if they're thermodynamically favorable, then they also don't really require ATP. Usually this is like a joining of a phosphate group onto a molecule without ATP. So ATP is often, in many cases, used to provide a phosphate group to a molecule. So that's whenever you get, ATP hydrolyzing into ADP and PI. Sometimes the chemical potential energy is used to fuse the phosphate group directly onto another molecule. So you see this in glycolysis, for example. But those are almost always in thermodynamically unfavorable events where they actually require energy to effectuate the process, as opposed to a thermodynamically favorable step, which it already has energy available that it can use due to the reaction producing it, that it can take a phosphate group from the purine pool, your fluid pool, and attach it to a molecule there directly. So it also just depends on the actual, they call it the delta G, the change in energy, whether it's thermodynamically favorable or not, a certain step. So it's, generally speaking, you're talking about thermodynamics and you're talking about concentration gradients as to when ATP is required or not. So anyway, yeah.
I'm staying at Mum's house and they've got a cat, and I'm slightly allergic to cats, and he's sitting just up there, and
yeah.
yeah.
My mom and my grandma are allergic to cats as well. My grandma's deathly allergic to them, actually.
I used to be a lot, like, very allergic to them. Like, I couldn't be in the same room as a cat, and I don't know, somehow over the years that's come right down, where just now, if I'm really close to them for a while, especially if I rub them and touch my face, it might set me off. But maybe that's a carnivore benefit too.
I was going to ask you. I don't know if it got better once he went carnivore. It wouldn't surprise
Yeah,
me.
I think it has. 'Cause it's only been the last 2 or 3 years that I'm not as allergic to them. Like, I wouldn't be able
Wow.
to be sitting here with this cat as well. So,
Huh.
yeah.
There you go.
Okay, so sort of taking on from that, again, something that I was taught, which you've spoken about, and I think Bart K speaks on about. We don't have three energy systems. We have one energy system with, I suppose, three gears or three cogs in the gearbox. Can you sort of, I suppose, do a quick overview of how that all fits together? Because the way I was taught, and the way I think is still taught many times, is like you've got your ATP PC system, which is like, for the first 10 seconds of a sprint or something really intense. And then you go into your anaerobic energy system for a few minutes where you're sort of producing ATP without oxygen. And then if you go over the 2 or 3 minute mark, you start needing to use oxygen and you go into your aerobic energy system. And it's not that at all, or not that simple.
Yeah. No. So in reality, we usually say 3 gears, but you really could say 4. One of them is just think of the fourth one's a really small one. With the other 3 are like same size, and then you got a small one just on the side or something. Here's what it is. Okay, so the first thing that you said about your phosphocreatine, you got like PCR, you got that. Phosphocreatine, that's true. PCR can function and does function as a buffer for energy demand that happens initially in exercise. But the best way of explaining it is to go through each of the 3 primary gears first, and explaining how they work together. So there are these 2 researchers, Shulman and Rothman, that I can't remember the exact year, but they discovered that they were intrigued by this observation that they were seeing, which was a production of lactate, which is colloquially considered lactic acid,
Yes, yeah.
In resting muscle activity. There is a certain level of lactate that is produced by muscles at resting usage. So like if I pick up this glass, pick up this pen, there is some lactate that's being produced. And they were confused because they're like, well, hold on a minute. The idea that we're taught is that lactate, this lactic acid, builds up during high-intensity training when you reach that anaerobic threshold.
Mm-hmm.
That's what you're taught. And so they thought, well, why is this being produced then? What's going on? And so they were like, well, is there low oxygen perfusion? Because that's also another case where lactate can be produced because, well, if there's low oxygen, hypoxia can produce lactate. And that's also another reason why it can cause lactic acidosis. If people are like low on oxygen, they've got ischemia, they've got a necrotic bowel or something, like dead tissue or something like that. You can produce lactate in other ways. But typically, if you've got perfect oxygen perfusion, normoxia as they call it, and you're at a resting energy state, then you're not going to be producing lactate. Well, what they found is that this persisted even in the context of normoxia, so they decided to look into it. Now, first what they did is they looked at a rattlesnake's rattle. This is in a study called Shaking Up Glycolysis. Yeah, they looked at a rattlesnake's rattle. And what they found was that even in all twitching of muscle fibers, at least in a rattlesnake's rattle at the time, they found that there was a degree of glycolytic activity, what's considered the anaerobic gear, even at the lowest intensities of muscle contractions. And this confused them. They went, hold on a second. That's not, that goes against the model that we're taught, like directly against the model, because you're not, this is supposed to be an anaerobic system. There's perfect oxygen perfusion to the tissue, and they're not engaging in high intensity training. Like there's no reason for this to be occurring. So what they did was they were like, okay, this is anomalous. Let's see if this translates to human beings. And so then comes a study that was called the glycogen shunt. And that is a study that proved that it happens in human beings as well, but it went deeper. And so there was, an aggregation of a lot of research that was done. And basically what they found collectively, mainly Shulman and Rothman, and then you've got also Brooks, which is an author that talked about PCR quite a bit. What they found was this. Literally every single muscle fiber of every single intensity, the slightest twitching was associated directly with glycogenolytic activity, glycogenolysis. That's the process by which your body breaks down glycogen, stored glycogen in your muscles to create ATP. You can actually look at this on a graph where there was a spike. So there's a level, you can see ATP produced from glycogen. It's a line here, and then it just spikes up and comes right back down. Spike. And it's within 10 to 40 milliseconds that this occurred. And the reason that there's an interval range is because it depends on whether your muscle fiber is fast twitch or slow twitch. So between 10 to 40 milliseconds, a muscle fiber would be completely drained of glycogen. Completely drained of glycogen. And so then the question becomes, okay, well, inferentially, what that means is that glycogen is required to power every single muscle fiber of all intensities. Like, why is it occurring? Why is the glycogen being stored in the first place in a muscle fiber and then being used for every single contraction of all intensities? So glycogen's fired, but then the question becomes, well, where is the body getting the ATP to resynthesize the glycogen for the next muscle fiber twitch? Well, that's where PCR and oxidative phosphorylation come in, your aerobic system. What is happening is that the muscle fiber, for every contraction, glycogen is being used to produce massive amounts of ATP per unit time, like a very high amount. And obviously we've covered how that ATP is being used to power contraction, sort of, because again, constructs and all that stuff, the mystery of that. But then in the mitochondria are operating at a steady rate, and they're producing chemical potential energy at ATP synthase. So, well, the energy that's being used to resynthesize glycogen, because you do need ATP to resynthesize glycogen, is actually being taken from the mitochondria via phosphocreatine. That's the middle gear. So think of aerobic metabolism, your mitochondria, as the bottom gear. Think of glycogenolysis as the top gear, and the middle gear, the linking gear, is PCR. It's phosphocreatine. Creatine itself is actually the molecule that's doing this. It has a very it has a very high high concentration gradient, and it's also very small in molar mass compared to ATP. So it's very quick. It can move across the membrane very quickly, as opposed to ATP, because ATP's concentration gradients are very low. It's all across the cell, all across the cell, ATP. And also, they're very large molecules relative to all the other surrounding molecules. So it's not that mitochondria can't export ATP. They can. In fact, there's it's ATP translocases, but there's molecules from plants. So plant toxin actually called attractylosides that inhibit these transporters, and they can cause death. And so it's not that the mitochondria don't do that. It's that it's extremely important that it takes that PCR. It's more efficient for creatine to take that energy. So creatine comes across, takes the energy from the mitochondria, the chemical potential energy, and it uses that energy to actually form PCR, phosphocreatine. So now there's chemical potential energy fused or sort of held between inorganic phosphate and creatine. And then it comes back around and it unleashes or relinquishes that energy to ADP and PI to form ATP, and then that ATP is used to resynthesize glycogen for the next muscle twitch. Those are how all three of those gears work. Now, PCR does function as a buffer on its own to be able to cause muscle fibers to twitch. It requires ATP, really, but glycogen is still used in every muscle fiber twitch of all intensities. So you're still using some glycogen.
Yeah.
Yeah. And then the fourth gear that I was talking about is whenever you get to the later stages of exercise, which is whenever you get a buildup of AMP. AMP is, well, it's created once again through high energy demand, where the demand outpaces the supply. So you need this demand stays here, the supply, it's slowly coming back down, right? So you get there's a fourth gear that kicks into gear and is activated when this occurs, and that is, I can't remember. I don't even know if there's a specific name for this, but what it does is usually you've got ATP hydrolyzing into ADP and PI. Well, what can also happen is if the rate of ATP production dips beyond the level that the body deems to be indicated at that time because of very high intensity training, persistent exercise to volitional exhaustion, then what you'll have is you'll actually have two ADP molecules bumping together to form ATP and AMP. So they'll actually form, it's like 2 and 2, you form 3 and 1, basically, is what you're doing. And that is considered to be a fourth gear. And the AMP building up is not a good thing because it perturbs the equilibrium, it perturbs the chemical pressures and all that, according to Le Chatelier's principle. And so what ends up happening is that as the AMP builds up in a cell, there's an enzyme called AMP deaminase, and it will form IMP and also an ammonium group, NH4+. They say ammonia, but in reality, it doesn't exist at the pH of the body. It's not, and the pKa value of it, it doesn't exist as that. This is ammonium. Ammonium is excreted. Usually it's used to create urea. So, and then that's reflected in blood urea nitrogen levels, but it's excreted in the kidneys and therefore in the urine. And then IMP goes through another process, and it goes on its own little journey. Okay, and if it's in the liver, IMP is actually used to create urate, which is colloquially considered uric acid. So those are the 4 gears of muscle energetics. This is why, for example, a muscle fiber will not twitch without glycogen. It won't do it. You will find, you can actually observe that when a muscle fiber is contracting, when a muscle group is contracting, only a certain amount of fibers in the muscle will be recruited for the lift, because obviously the body's efficient in that respect. It's not going to use more than it has to. But during the course of a lift, you'll actually see that once the initial muscle fibers for the first sort of collective contraction to lift a weight up off the ground, once those have twitched, they will not twitch again until they refill with glycogen. Think of like an air gun or something that pop, shoot balls out. So therefore, you'll actually see that certain muscle fibers will twitch, and then as you continue to lift the weight up, other muscle fibers will compensate. Others will be recruited. It's like a rippling effect.
Yeah.
That's why you see that, because a muscle fiber will not twitch without that glycogen. It's one of the other reasons why you will see that one of the common denominators for the pain that you feel during the course of a lift, that burning sensation, it is directly concomitant with a decrease in blood glucose levels, actually. right at the very end, you'll actually see the right at the very, like the burn of an exercise, at the very like last bit of the burn, you'll see that there's a dip in blood glucose, and it comes right back up. And it seems to me that it's because now what you've done is you've outpaced the sort of rate at which your body can produce the ATP for the energy demand. That's what you've done. It's also a self-protective mechanism. Like I think that's the primary thing that it is, a self-protective mechanism, because there are micro tears of myosin chains during an exercise, if you push them hard enough, which is why you can seriously damage your muscles if you, I don't know, have an adrenaline rush, and you overdo it in that respect. Adrenaline has a numbing effect in many ways. Like there's inhibitory effects on things, and that's why you can get these ostensible superhuman powers seen, but they really harm people.
And people lifting cars off kids and stories like that. Is that sort of, I suppose, part of the reason why you can only sprint at your fastest for 10, 12 seconds, and you can't do it for 30 seconds or a minute or 3 minutes? Because if you were to sprint that hard for that long, you would cause too much damage.
Yeah, that's one factor, I think. I mean, obviously, there's also the factor of just, VO2 max. Obviously, if you're not trained for it, like, you're going to just perform less time, and your cardiac health, because, there's an endurance level, there's some stamina there involved, just like anything else. I mean, but yes, there, even for the most active athletes, like the most adept at that field and that exercise, and that skill, really, they in reality could go a lot longer, but the body doesn't let them, because that's just not, there. I was, again, talking about that Anthony Chafee conversation that I had, the conversation that I had with Anthony. He mentioned that, I can't remember the name of it, but that burning sensation, that that failure that you end up having, you can actually, it's really interesting, you can override that temporarily if you just put something sweet on people's tongues.
Yeah, I've heard him talk about that. Yeah.
So that also signifies, it tells the body, okay, something sweet's coming.
You don't even need to ingest the glucose, just that sensation that your body thinks it's coming in.
Yep.
Does your body think? No.
Right. But it signals it to the body. It's like, okay, so we're going to be getting glucose, and so we're going to be able to go a little bit longer. So there's some holes in the picture, because if you're going to believe that it's a self-protective mechanism, then why would it be the case that if the body thought there was more glucose coming in, that it would continue the exercise? Because, well, if it's purely self-protective, it shouldn't matter what you ingest during the course of the exercise. It's like, so clearly it's a fuel partitioning thing as well, which is why I said in the beginning, it was the first thing I said, which was, well, it's the glucose thing. Like, you're outpacing, and Tim Noakes did the study as well on like 10 people, I think. So again, vast limitations of the study. I'm not pretending otherwise, but there was a interesting observation, which is that these people that were given an actual, a literal shot of glucose, but it was like a very small amount. You're talking like between 2 and 5 grams. It wasn't even 5 grams of glucose. There was a statistically significant increase in their performance output as compared to when they didn't do that. Like, it was interesting. And so I also think it is an actual fuel partitioning issue as well. And the question then becomes, should you override it though?
Yeah.
Like, I don't think you should, but
And that would sort of, I suppose, be similar. Like, why do people have a 1 rep max? So in a trained individual, say, let's take an exercise like a squat or a bench press. You've got that maximum weight, or that weight that you can lift only one time. Like, why can't you lift it a second time? If you can lift it, why can't you then lift it a second time?
Right. And it's clear to me that it is a fuel partitioning issue. Like, you've got, so when you lift that 1 rep max, like the weight, you lift that weight in 1 rep max, you're employing multiple muscle fibers all at once. They're all firing. Like, the energy requirement is definitely going to much more quickly outpace the supply and the rate of the supply of more energy, because again, you got to think of it in the context of the 4 gears. If you like, for example, here's something else. There's a lot that I actually didn't even cover in those 3 gears or those 4 gears. Like, for example, the reason that we have glycogen in the first place, I already covered it, which was that you require the glycogen because the amount of energy that is demanded from an individual muscle fiber, even for the lightest bit of activity, is much more than the mitochondria can supply at its normal resting rate. However, during the course of an exercise, your mitochondria, over the course of 3 minutes, actually speed up the rate of production of ATP production to a rate that eventually does meet that demand. Doesn't change the fact that you're still going to be using glycogen, but it will actually reach that demand. It just takes a minute. It also takes 3 minutes to slow back down to its normal rate.
So the maximum rate is reached after 3 minutes.
Yeah, of high intensity training. Yep. So if you're after 3 minutes, which also that's what explains the excess post oxygen consumption, also known as EPOC.
EPOC, yeah.
Yeah, after exercise. It's very acute. I know that a lot of people will try and say EPOC. Well, that allows you to you can eat more if you exercise more because of EPOC or something, because you're oxidizing more substrate. It's like, yeah, for 3 minutes, and then it goes right back to normal. Like, it's not. I reacted to someone that said this, and I was like, okay, that's a huge reach. But that's something that happens. But also, why does your body not use glycolysis instead of glycogenolysis? Which, first of all, people might have a question with the question because they go, well, isn't it just called muscle glycolysis? Like we refer to it as muscle glycolysis. Well, it's not precisely glycolysis. They're actually 2 different pathways. Glycolysis requires 2 ATP to be expended, one in the first step and one in the third step. Glycogenolysis requires 1 less ATP because glycolysis is lumped into glycogenolysis. It's actually a subset. It's subsumed by the overall pathway, glycogenolysis, and it requires 1 less ATP. And so it's efficient. It's more efficient for the body to do that than it is for it to depend on. It's impossible for it to depend on purely mitochondria. So this also explains why this isn't a human exclusive phenomenon. It explains why it's for all animals, because, well, animals have mitochondria in them, and they're not going to if any animal moves and uses muscles, and even the lightest bit of movement in muscle fibers requires rapid amounts of ATP production, then they're not going to be able to depend on their mitochondria for it. They're going to have another system, and that happens to be glycogenolysis. So, or at the very least, like glycolysis. I don't know if, but no, it's glycogenolysis. Yeah. So, anyway, I can't remember why I went to talk about it again. What you asked me something that I started talking about that.
Yeah, I was asking about 1 rep max and why
a
1 rep max can't be a 10 rep max or.
Yeah, I mean, really, it goes. If you're going to zoom in on all of those 4 gears and then zoom back out to get a very broad look at like what's actually going on. Like, if I think of it as like pistons firing, you're going to need more piston activity to get the thing up. So it's usually you do it slower. That's why. And then whenever you get it up, you can usually, in many cases, you sort of hold it up. But that's if you lock your arms out, which you're not supposed to do, especially with super high weight, because then you're putting the load on your joint. That's the only reason why you can keep it up, because now you've unloaded the muscle. You're loading it on your joint, which, oh boy, yeah, your joints are strong. Yeah, but they're only so strong. And then you come back down. Well, you're not coming back down because that would be a second rep. You'd be going for a second rep. But yeah, you do it slower, and you can't really do a second one because even when if you were to go do a second one, when you're going back down, you're still recruiting your muscles.
Yeah.
Because if not, it would just fall on you. So I mean, it's just an outpacing of energy supply to energy demand. That's really what it is. Like, even just during the course of a lift, that's what's going on. You're reducing you're increasing the amount of energy, and you're making it energy demand, and you're making it outpace the rate at which your body can supply the muscle fibers for more twitches. This is also why Once you perform an exercise bout or a set of something, you can put the weight down and then you can come back about 30 seconds later and lift it more. Now, eventually, it's still going to be diminishing returns, absolutely, and also this. I mean, it fits the model though, because the diminishing returns matches the resting glycogen status after you have glycogen depletion for like 24 to 48 hours after an exercise in the muscle fibers that are targeted
specifically.
Yeah.
So if you have a big leg day, they're depleted from your leg muscles. Mm-hmm.
And it also that coincides directly with a decrease in the amount of force that you can put out. So it directly fits the model that I'm talking about. You'll have slow replenishment of that glycogen back. And of course, again, diminishing returns. You get most of it, I think, in the first 24 hours, and then completely after about 48. Again, first order kinetics to zero order kinetics on the graph. Anyway, it directly fits that model. But yes, whenever you talk about, people talk about aerobic exercise versus anaerobic exercise. Aerobic exercise is exercise that doesn't employ large amounts of glycolytic activity, glycogenolytic activity, really. It's like the walking, sometimes swimming, but even then swimming can recruit a lot of muscle. But the most common one people think of is like walking or
a slow jog.
Yes, or a slow jog. Whenever you get to the higher intensity, you're using more muscle. So of course you're going to be using more of the anaerobic gear. People think it's a threshold, like you shift an entire gear. It's like, oh, this is a system that you're in, aerobic system. Now we need to switch over to anaerobic system. You reach a certain threshold. It's like, if that's the case, then why is it the case that during anaerobic exercise, that's the exercise that oxygen consumption is maximized? You're breathing the most oxygen. You're using the most oxygen during that. That's why you measure a VO2 max during high intensity training.
Yeah. Like,
so it doesn't make
nothing gets my breathing and my heart rate up than it's either sprinting or lifting really heavy, or as heavy as I can.
Exactly. So it's based on a fallacy that they're independent gears, when in reality it's one system and it's multiple gears working in tandem. In fact, they actually resemble a capacitant, like an electrical circuit with capacitance. Mm-hmm. Because they're all connected to the same source, oxidative phosphorylation, the mitochondria. That's where the energy is coming from. Ultimately, glycogen it's a compact form of energy that has energy in it that was taken from the mitochondria at one point. So, glycogen, you got a lot of glucose polymers, and then when it breaks down and forms all that ATP, it's like, but where did that energy that was used to synthesize the glycogen ultimately come from? It came from the mitochondria. It's just they store it in a packaged form for immediate usage until it needs to replenish it again. And then, so if you think of all your muscle fibers as different pistons, you'll fire like some pistons for the initial phase of a lift, and then during the rest of it, other pistons fire until the other pistons that fired first replenish, and then they fire again. It's just that's why you see the muscles rippling like that.
I like that analogy.
Yeah. So it's a very complicated picture, but it becomes easier to understand. I mean, I haven't gotten to this in my biochemistry course yet for my early access members. We'll get to muscle energetics way later, but I'm opportunistic about it and hopeful about it, and I'm looking forward to it. And it's also very daunting at the same time, because trying to make this picture of what's going on, it's very, very, very difficult. Because I know it in my head, but actually putting it, realizing it and making it materialized on a graph or something, it's difficult. So it's going to be challenging, but I'm looking forward to it nevertheless.
Yes, I'm looking forward to.
Yeah, good.
It too, once it's out.
Yeah.
Are we sort of reaching time?
Yeah, I got 5 minutes. So look. Well, the thing that I'll say is that we can do another. That's what I say to all the people that I talk to, because I like doing this stuff. I like talking about it. So.
Yeah, because I suppose the questions I'm asking you are not like 10-second answers. They're like 20 minutes.
No. Yeah.
And even then, you're condensing a 3-hour lecture into a 10 or 15 minute sort of quick conversation.
Yeah. I mean, the questions you're asking are about stuff that I really like going into anyway. So especially the muscle energetic stuff. It's very, very fun to think about that. Also, like acid base balance. I don't know if you have any questions about that, but that's
Yeah, I've sort of, yeah, that sort of, I'm at the point where that still does my head in a little bit when I think about it, because I did chemistry in high school, but it's been a long time since I've
yeah.
had to sort of think about it at that level. So, yeah, that whole sort of series of to and fro between Bart and what's his name?
James
That
DiNicolantonio.
yes, that's the guy that we're all going to dissolve our bones because we eat too much meat and we don't drink bicarbonate. Um,
Yeah,
yeah, like
baking
I,
soda.
I've listened to all the lectures and it was very interesting, but like in terms of the understanding the chemistry behind it, it's very superficial level for me at this stage.
Yeah,
But
yeah, yeah. I
I
mean,
mean, biochemistry involves a lot of chemistry, obviously, so I've got to
yep,
relearn all that.
yeah. The major thing about it is, I mean, you do have to look into like electro negativity and stuff. Like, the periodic table and stuff, and then you've got the acid base balance. I think acid base balance is actually the biggest, most relevant factor. Yes, of course, you've got like when it comes to the affinity for certain um, reactions to occur, you're going to look at electronegativity. But I mean, I feel like it's relatively simplistic, but again, that's also coming from me, and I don't even I mean this in the most humble way I can configure this. I like obsess over this, so I'm like I kind of I kind of just get it, but a lot of people they don't. I mean, that's just the fact. So it's not to say that you're one of those people. It's just some people might listen to it and say, "Oh, well, good for you, Eddie." It's like I don't mean it in an insulting way, like,
No, no, I
but
understand. I want to get it. As I said, when I start doing something, I
well,
want to learn as much about it as possible. So,
what I'll say is acid base balance. That whole concept took me in order to fully grasp it. I would say that the first like 70% of it I got within a month. The rest of the 30% probably took me about 5 months. So it took me a long time to fully understand it. And now I made sure to get to the point where I was going to understand it to the point where if someone quizzed me on it 2 years from now, I'd be like, oh, yeah, yeah, yeah. Yeah, yeah, I know this whole thing. Like, I needed to make sure of that. And that was part of why it took me 5 months. I mean, to get a general gist of it, I don't think it would take forever, but, yeah. Yeah, yeah, yeah. Anyway, that's I get that's all the time I have today. But we
that's
can,
fine. Thank you for it.
yeah, yeah, absolutely. Look, just email me. It was good talking to you. Email me whenever you have some more time to talk about ask your questions and stuff, and then we'll schedule another one as soon as possible. But yeah, Nick, it was good. It was good speaking with you, and I'll talk to you later.
It was great talking to you as well. Have a Merry Christmas and great New Year.
Yes, yes, you too. Yeah, I'll talk to you later.
Okay, cheers. Thanks.
Yeah.
Bye.